Hello, Mr. Allman here, and this is your Allmancast on cell transport processes. As you now know, the cell membrane is made up of a phospholipid bilayer with embedded membrane proteins. That phospholipid bilayer has the main role of being an effective barrier, keeping good things inside the cell and allowing for the elimination or the exclusion of things that are harmful or dangerous. So, if the cell membrane is acting as a barrier, how do good things get in, and how do those bad things get out? In this video, I want to walk you through the overview of active and passive transport and how it is that these different methods are necessary in order to get different things into and out of the cell. Now, the transport of materials is really an application of chemistry and physics, because all things being equal, matter, meaning things, will spread out. This is based on the second law of thermodynamics, that idea that the universe is constantly becoming more disordered. And so there is a constant force to be able to move materials from where they are more concentrated to where they are less until equilibrium is reached. Now, cells will utilize this force in passive transport. Passive transport requires no energy, and it's something that's going to happen entirely on its own as material moves from a higher to lower concentration. Now, diffusion is going to be the movement of that molecules that we can see and describe from where they are more concentrated to where they are less as they are spreading out. And diffusion is going to be the movement across a semipermeable membrane, such as the plasma membrane, the mitochondrial membrane, or the nuclear membrane. Now, as long as a molecule can fit through that membrane, diffusion is inescapable. If you see in the image here, you have a low concentration of sugar on the left and a higher concentration of sugar on the right. Now, what's going to happen? There's going to be a movement across that barrier. Now, if the sugar molecules are able to, they will move from the right to the left. However, as you can see, each of those molecules of sugar is too big to fit through the pores in that membrane. So, water is going to want to move from where it is more concentrated on the left to where it is less concentrated on the right. And so, without the application of any heat or energy, there's going to be movement of water from the left to the right, as you can see illustrated by the increase there in volume. This movement of water across a semipermeable membrane is also called osmosis. And osmosis is going to be the movement of water in the opposite direction of the solute. So whereas the higher solute concentration in this picture is on the right, and that you expect that sugar to move towards the left, water will actually move in the opposite direction, from left to right. So we could define the movement of water by osmosis as the movement of water towards an area of greater solute concentration. The movement of water towards an area of greater solute concentration. In reality, diffusion is not happening in one direction, but molecules are constantly moving in both directions across a membrane, assuming that they can get through. Now, in the picture on the top, you can see the circles here represent a solute. And so the solute is highly concentrated on the left in comparison to the right, which is pure water. So there's going to be movement of those solute molecules from the left to the right through the membrane. However, the molecules are constantly in motion, and so even after they're equally distributed on both sides, there's going to be movement of some of those molecules to the left across the membrane and an equal number of molecules to the right across the membrane. In equilibrium, the concentration of these solute molecules is equal on both sides. However, they are still moving through that membrane. If we add in a second solute, you can see that the solutes will move independently of each other. So in this case, the orange dye has a net diffusion from the left to the right because there is no orange dye on the right, and so that orange dye is moving towards equilibrium. Simultaneously, the purple dye, which begins in high concentration on the right, is going to be moving towards the left. And when equilibrium is reached, there will be an equal concentration of both colors on both sides. the movement of those molecules still continues, because as long as that membrane is permeable or allows the movement of them, they will continue to move in both directions. However, at that point, we would no longer notice any change in color because there is no net movement. The movement in one direction equals and cancels out the movement in the opposite direction. So, as I said, the principles of chemistry here also need to be considered. We need to think about what can diffuse through the bilayer, because not all molecules can move through the membrane. We call the membrane semi-permeable because it only allows some materials to move across. Well, the physical and chemical properties need to be considered because the size of molecules is important. Small spaces exist in between the molecules of the membrane, and so large molecules are going to be physically blocked. Large molecules, things like glucose, sucrose, proteins, DNA, even amino acids, any large multi-atom molecule is likely to be blocked because it's simply too large to be able to fit across if it's not only 1, 2, or 3 atoms large. Some of those small molecules that do fit are things like gas molecules. Oxygen is made up of 2 atoms. CO2 is made up of 3 atoms. Nitrogen is made up of 2 atoms. Those molecules are small enough in size to be able to actually fit through those spaces in between the phospholipids. However, size isn't the only thing that matters. In addition, chemical properties are important because the interior of the membrane is made up of fatty acids and thus is hydrophobic. Because it is hydrophobic, anything that is polar or charged is unlikely to be able to move across because it's going to be repelled by that hydrophobic interior. So again, charged molecules like sugar molecules, amino acids, ions like chloride, potassium, sodium listed here, anything that is polar or charged is not going to be able to move through. Something that is chemically hydrophobic, like a fat, is going to be able to diffuse in that membrane, so that would be the exception. Something that is large but hydrophobic is still going to be able to dissolve into and move across that plasma membrane. Now, what about all that other stuff? What about those proteins, those sugar molecules? Those things still need to get into the cell. Well, in order to get in, they need help. So that help comes in the form of those transport proteins, which are embedded in the membrane. And those transport proteins are going to allow the movement of charged or polar or larger molecules across from outside the cell in. In the 2 examples that you see here, you can see an aquaporin on the left. This is a water pore. This is going to allow water molecules to move from the exterior of the cell inside or from the interior of the cell out. You have an ion channel, which similarly is an embedded transport protein, which is going to provide a channel through which an ion can actually move across. These channels are going to be specific to a solute or solvent. So an aquaporin will allow water, but it doesn't allow other materials to move across. An ion channel might be specific to one particular ion, like a potassium channel or a sodium channel or a chloride channel. So the shape and size and the chemical properties of the interior pore there are going to specifically match the size and chemical properties of the thing it's trying to move. You can see there are 2 main ways in which these transport proteins might allow materials to move across in the process called facilitated diffusion. Facilitated diffusion is passive transport because it requires no energy. It's going to be the movement of materials from where they are more concentrated to where they are less. There could be a channel protein which provides a pore, and that pore will just simply allow materials to move through it by diffusion from where they are more to less concentrated. There's no control over this. It simply provides the opening, and so that opening molecules will move through randomly. Carrier proteins are going to be slightly more sophisticated because carrier proteins are going to be unidirectional. So as you can see in this case, the blue sphere, the carrier protein will actually shift. It'll open at one end, allow that blue molecule to attach, and then it'll transport that protein across when it opens in the other direction. So diffusion will happen if the blue molecule is more concentrated on the outside than in. It's going to allow for the movement of that material in. However, it's not going to allow those blue molecules to move back in the opposite direction. So no energy is required, but there is a change in shape here in that carrier protein as the diffusion occurs. The example of an important molecule that is transported by these carrier proteins is glucose. And you can understand why it is that the cell would not want to allow glucose to diffuse back out. Glucose is used as a source of energy, and so once that glucose diffuses in, the cell is going to hold onto and keep that glucose as a source of energy indefinitely. And under no circumstances is the cell going to want to lose and give up that glucose back to the exterior of the cell, even if the bloodstream on the outside has a very low sugar concentration. Now we move to the other form of transport, which is active transport. So this is going to be the opposite. As opposed to the movement of materials from a high to low concentration, active transport requires energy. That energy is required to cancel out that universal property that things spread out and equilibrium is reached. So we have the movement of materials from where they are more highly concentrated to where they are more low, to a low concentration. This process has to use a protein, because if there are only pores or openings in the membrane, then the movement is always going to be in the opposite direction. It'll be in the direction of diffusion. So proteins, however, can be controlled and manipulated in order to allow this movement in the opposite direction of what you would expect. If you look at this diagram, this shows a very common process using a proton pump. A proton pump, represented by this purple shape, is going to move protons, which are a single proton. We also call them H pluses because it's a positively charged hydrogen atom that has lost its electron. There are a lot of circumstances in which these protons want to be concentrated on one side of the cell, for example, outside of the cell, to create a form of energy that could be used for reasons that you'll see in a moment. Energy is required in the form of ATP, and you see that the concentration gradient has been set up. There is a high concentration of protons outside the cell and a low concentration of protons inside the cell. However, using energy, whatever protons are left can still be moved and concentrated on the exterior of the cell. So this is in the opposite direction that diffusion would allow for the movement of these protons, but moving them on one side of the cell, as I said, can be useful for situations like you see here. If you set up a concentration gradient of a lot of protons on one side of the membrane, those protons can be allowed to diffuse back in. So the proton pump pumps the protons out. By diffusion, those protons, they want to come back in. And if the cell lets those protons, if it gives them a channel to move through, they're going to do so. And so a co-transporter can be used to simultaneously allow the diffusion of those protons back in, along with a second molecule that the cell really wants. For example, sucrose, which the cell can use as a source of energy. So use energy to pump protons out, and then when the cell allows those protons back in, use the movement of those protons to bring along a second molecule that's very helpful. The usual molecule that will provide the energy for active transport is ATP. ATP is a large molecule that has 3 phosphates on the end of it, the same phosphate that we find in the phospholipid of the plasma membrane. Now, there is one little part of that ATP molecule that can be broken off in a process called phosphorylation. And when phosphorylation happens, that can change the shape of the protein. And this can be a way to be able to accomplish active transport. Now, the sodium-potassium pump is probably the most important example that you should know of active transport by ion pumps. Again, ion pumps, the goal for them is to try to concentrate ions on one side of the membrane or another. For neurons, which use these potassium pumps and these ion gradients to be able to allow communication through the body, what they are trying to do is set up a situation where there is very little sodium inside the cell and a lot of sodium outside the cell. Conversely, they want a lot of potassium inside the cell, even if there is very little potassium outside. So how do they do that? Well, it starts by the opening of the ion pump towards the interior of the cell, and there are spaces where sodium is going to be attracted to and bind to that proton, to this sodium-potassium pump. Then ATP is going to be used to change the shape of the protein. Now the protein is going to close to the interior of the cell, and it's going to open to the exterior of the cell. And after it changes shape, those sodium ions are no longer as attracted to it, and so they're going to leave. They're going to leave and exit the cell. Now, there are spaces exposed that potassium ions outside of the cell are going to be attracted to. So the potassium binds. That phosphate, which was left behind from the ATP, is going to get broken off. And once it's broken off, it's going to change the shape of the protein again, closing it to the exterior of the cell and opening it to the interior of the cell, where those potassiums, again, will be ejected from the protein. And if this process repeats again and again and again, what you'll see is that more and more sodium will be concentrated outside of the cell, and more potassium will be concentrated inside of the cell. Those concentration gradients are then used by neurons to accomplish something called an action potential, which we'll learn about later in the course. And that is going to be how cells can communicate within the body. As always, if you have any questions, please let me know. Thank you for listening.